Hello everybody, my name is Charles, and I am a cell of Chlamydomonas. Without further ado, let's get right into this video. To understand how cells function, it is important to first understand how they regulate their internal environment. Every cell is constantly interacting with its surroundings, taking in materials it needs and removing waste products it no longer needs. All of this controlled movement happens through a boundary known as the plasma membrane. The plasma membrane surrounds the cell and acts as a selective barrier. This means it does not allow every substance to pass freely in or out. Instead, it carefully controls what enters and leaves depending on what the cell needs to survive and function properly. This control is essential for maintaining stable internal conditions even when the external environment changes. This stability is called homeostasis, and nearly every transport process in the cell is connected to it in some way. The plasma membrane is mainly made of a structure called the phospholipid bilayer. A phospholipid is a special type of molecule that has two very different regions. The phosphate head is hydrophilic, meaning it is attracted to water. Because of this, it faces both the watery environments inside the cell and the watery environment outside the cell. In contrast, the fatty acid tails are hydrophobic, meaning they repel water, so they point inward away from the water, forming the interior of the membrane. This arrangement creates a stable double layer that forms the basic structure of the membrane. The membrane is described by the fluid mosaic model. It is called fluid because the phospholipids and many embedded proteins can move sideways within the layer, giving the membrane flexibility. It is called a mosaic because it is made of many different components working together in one structure. These components include proteins, cholesterol, and carbohydrates. Proteins are responsible for many functions, including transporting materials across the membrane and receiving signals from other cells. Cholesterol helps maintain membrane stability by preventing it from becoming too rigid in cold conditions or too fluid in warm conditions. Carbohydrates are often found on the outer surface of the membrane and are used for cell recognition, allowing cells to identify each other and communicate properly. Together, these features make the membrane both flexible and highly functional while still maintaining control over what passes through it. Movement across the membrane occurs in two major ways: passive and active transport. Passive transport is the movement of molecules without the use of cellular energy. In this process, molecules move naturally from an area of high concentration to an area of low concentration. This movement down a concentration gradient happens because molecules are constantly in motion and tend to spread out evenly over time. One form of passive transport is simple diffusion. In simple diffusion, many nonpolar molecules such as oxygen and carbon dioxide are able to pass directly through the phospholipid bilayer. They do not need any assistance because they are small enough and do not interact strongly with the hydrophobic interior of the membrane. However, not all substances can cross the membrane this way. Larger molecules or charged particles such as glucose and ions are blocked by the hydrophobic core of the membrane. Because of this, they require help to move across. This leads to a process called facilitated diffusion. In facilitated diffusion, transport proteins assist molecules in crossing the membrane. These proteins can act as channels that form pores through the membrane or as carrier proteins that change shape to move substances across. Even though proteins are involved, no energy is used, and molecules still move from high to low concentration, which is why this process is still considered part of passive transport. A specialized form of passive transport is osmosis, which refers specifically to the movement of water across a membrane. Water is a small molecule that can move through the membrane, but in many cells it primarily moves through specialized protein channels called aquaporins. Water movement here is driven by differences in solute concentration, meaning it moves towards areas where there are more dissolved particles. To describe how cells respond to different environments, we use three terms: hypertonic, hypotonic, and isotonic. In hypertonic solution, the surrounding environment has a higher concentration of solutes than the inside of the cell. Because of this difference, water moves out of the cell. And as water leaves, the cell loses volume and begins to shrink or shrivel. In a hypotonic solution, the outside environment has a lower solute concentration than inside the cell. The water moves into the cell, causing it to swell. In animal cells, this can lead to bursting, while in plant cells, the cell wall prevents bursting and instead creates turgor pressure, which makes the cell firm and rigid. In an isotonic solution, the solute concentration is equal inside and outside the cell. Water still moves in both directions, but at equal rates, so there's no net movement. This creates a stable balance known as dynamic equilibrium. Active transport is the opposite of passive transport. Instead of moving substances down a concentration gradient, it moves substances against the gradient, from an area of low concentration to an area of high concentration. Because this movement is not natural, it requires energy in the form of ATP. Active transport uses specialized proteins called pumps that are embedded in the membrane. One of the most important examples is the sodium-potassium pump, which is essential for many cellular processes, especially in nerve and muscle cells. When ATP binds to the pump, it causes a chemical change in the protein structure. This change, called phosphorylation, causes the protein to change shape. As a result, ions are physically moved across the membrane through their concentration gradient. This process is extremely important because it allows cells to maintain specific internal conditions, even when the external environment is very different. This regulation is a key part of homeostasis. Some substances are too large to pass through membrane proteins at all. In these cases, cells use bulk transport, which also requires energy. In endocytosis, the plasma membrane surrounds a large particle or substance and folds inward until the membrane pinches and forms a vesicle that brings the material into the cell. In exocytosis, vesicles inside the cell fuse with the plasma membrane and release their contents outside the cell. This is often used to remove waste or to export important molecules like proteins. Overall, cell transport can be divided into passive and active processes. Passive transport moves substances down their gradient without energy, while active transport moves substances against the gradient using ATP. Together, these processes allow the cell to carefully control its internal environment and maintain homeostasis under constantly changing conditions.